As theories of cortical organization and function become more sophisticated, neuroscientists have started piecing together different types of data in order to understand how brain regions communicate. But one emerging idea is that the brain is organized in a hierarchy, in which feed-forward signals from early sensory areas are exchanged with feedback signals from higher cortical regions. Now, one question is what such signals look like, and whether they are communicated via dedicated anatomical pathways. Recent work indeed suggests that much like opposing lanes on a highway, feed-forward information is predominantly carried via deeper cortical layers, and feedback information is carried via superficial layers in cortex. But in addition to this, it has been proposed that the activity in these feedback and feed-forward channels has distinct characteristics. So while feed-forward activity occurs mainly in lower frequencies, such as alpha and beta ranges, feedback activity is mainly conveyed in higher frequency ranges. However, so far the evidence for these ideas has come from invasive recordings in non-human primates and indirect measures of neural activity in humans. Now, there are currently no ways to test these ideas in the human brain non-invasively and over the whole brain because of technical limitations. The ideal candidate for addressing this would seem to be magnetoencephalography, or MEG for short. This technique measures brain activity directly and non-invasively, and it covers the whole brain. But until recently, its spatial resolution has been limited. This is because subjects typically move, and because precise knowledge of the brain's location is often inaccurate. So much like a microscope whose lens is out of focus and whose underlying probe moves, it limits the precision with which we can assess these ideas with MEG. Now, our group has recently developed these funky-looking foam headcasts, which are individually made for each participant, so the inside is a perfect mold of the participant's head, and the outside fits perfectly inside the MEG scanner. And we have shown that this seemingly simple solution reduces head movement during a scan to within 1 to 2 millimeters. It also ensures that every time someone gets in the scanner, their brain is in exactly the same place. Now, before we use these headcasts with human subjects to probe the organization of feed-forward and feedback pathways, we wanted to determine if we could, in principle, tell whether measured MEG data came from deep or superficial cortical layers. So to do this, we needed a data set where we already knew the ground truth. So we created some simulated MEG data using a generative or forward model of how electrical activity in different parts of the brain generates tiny changes in the magnetic field that we can measure from outside of the head with MEG. So we simulated activity in different parts of the brain in either deep or superficial cortical layers and used the forward model to create different simulated MEG datasets. We then tested two types of analyses in terms of how well they could recover the location of the simulated signal in these datasets, so whether it was coming from deep or superficial cortical layers. Now, the first type of analysis gave a global picture. It told us over the whole brain was activity occurring in more deep or more superficial layers. This type compared two generative models in terms of how well they matched the data. One assuming that all of the activity was occurring in deep layers, and the other assuming that all of the activity was occurring in superficial layers. If the deep layer model matched the data better, then we can assume that the activity was coming from deep cortical layers, and vice versa for the superficial layer model. And the second type of analysis allowed us to zoom in on particular brain regions. It used a generative model that combined the deep and superficial layers and estimated the strength of the activity on each. We then restricted our view to where the activity was strongest across both layers, and within this region compared the activity between the two layers. And we found that both types of analyses were able to accurately reconstruct where the simulated activity originated from, whether it was in deep or superficial layers. And importantly, both types of analyses were able to do so even when the data was quite noisy. Now, this is exciting because it means under certain conditions, the type that we think we can achieve with these headcasts, we can tell whether brain activity is occurring in deep or superficial cortical layers. So now we can finally test modern theories of cortical organization in the human brain, and we can do it non-invasively and globally, covering the whole brain. So stay tuned, because in future studies we set out to do exactly this.